Good morning.
Good morning, sir.
How are you guys?
Right, sir.
You're enjoying your days in the house? Quite boring sitting in the house, right? Okay. Shall we go to the topic of discussion today? So we have a topic for discussion is molecular techniques. Now, what is the importance of this molecular techniques? Let us discuss for some time. Now, as the science is growing, the medicine also is growing. As the medicine grows here, that we need to go to the molecular level to teach a particular disease. For that, I have the molecular techniques which are involved in the subject of microbiology. I have a molecular techniques which is being used in the subject of pathology. I have a molecular techniques which is being used in the subject of biochemistry and molecular physiology. Now, why do I want to know this molecular techniques? Now you can see here as the day passes, time changes. We have a day-to-day change of entry of the new viruses, new antigens into the market, into the world, and which will invade our bodies, bringing about certain reactions to the bodies. Now, so these particular antigens are which they invade our body, we become the host for them. So we had last time discussed when I was teaching you about the immunity, about an antigen. The main property of an antigen, if we quickly recap it, it should have the two properties. One property, which what I call it as, it will show a reactivity. It will show a reactivity. This is the one point which is very important for us. Every antigen, when we say it's a specific antigen, it will show the reactivity. That's the point number one I need to understand and remember and realize that, because when it invades the host tissue and then it need to survive, when it need to survive, it has to show a property called as an reactivity. That's one important fundamental point and the property of an antigen. Now, what is the second point which is very important for us, that when any antigen which will invade the host, the person or the host or any host, whether it's an animal or a human, so that it has to bring about an immunoreactivity. This is very important point I need to understand. It should bring about immune reaction in the host. Then only I call that one as an antigen, perfect type of an antigen. Now, this is one fundamental point I need to concentrate on to that. We call it as an reactivity. So that it will show certain reactivity in the host cells. So that reactivity substances which are released from this particular antigens bring about the mortality or the death of the patient or death of a particular part of the tissue. It will bring about an adverse effects into our body. In order to stop this reactivity, for us it's very important to find out that which type of the technique should we adopt to identify that particular antigens, to treat it, to stop its reactivity, so that the molecular techniques are going to be useful in this. Now, what is the main molecular techniques? We have the plenty of molecular techniques we are going to learn. Part by part, I am going to teach you about these molecular techniques. Today, we are concentrating much more onto this, what we call it as a report. You could have seen this. It is being generated by the computer with one technique called as a molecular technique, We term it as an RT-PCR. This technique, what you can see here, we call it as an RT-PCR. Now we call this one as an PCR is the basic technique actually. And then they adopted, and as the improvement is gone, and then they have started adding so many things that we are going to discuss is after some time. Then now, so this is exactly what we call it as a report. So being a medical doctor, when the person come with this report, I should be able to analyze what it is presented in this particular report. What it is been presented here. It's very important for us to learn and interpret this particular report. What it says that we have done the test for the SARS COVID-2. It means that SARS is a virus that was invaded our body or to the host of the humans, but a new type of the virus which invaded nowadays, which belongs to the category of an SARS, we call it as an COVID-2. COVID-2. So we call it as an SARS-CoV-2 and that too COVID-19. Did you see here that as the virus invaded into the body of the human? So we have given the name as an COVID-19. And nowadays, I see any channel and any newspaper, any social media platform, everyone have a bombardment with this, what we call it as a COVID-19, have a mortality of this one. We don't know which type of the treatment we are going to give it. Is there availability of the vaccine? If the availability of the vaccines is there, which type of the vaccines we are going to use it here? It is attenuated or an augmented vaccines, or we have a different types of the vaccines. So our recombinant DNA vaccines, RNA vaccines. We have plenty of the things which you are going to learn today's biochemistry class about that. So how do I read this report when it is in my hand and it is when it is presented to me? I generally look, being a normal person who was not associated with the medical field. I just look at this one point which I call it as it is positive. Then blindly I'll say that, hey, you are a COVID positive person. But I need to do an analysis. How do I do analysis? The analysis will be done if I know the technique very clearly. If I don't know the technique very clearly, blindly I depend upon the English literature called positive and negative. But I should not depend upon because I'm in a medical. I need to learn the things which are coming into our diagnostic field now because the diagnosis plays a very important role in treating the patient. I should know that when I call it as a normal, when I call it as a severely affected, when I call it as moderately affected, can I give an intervention at this time? I can use these techniques as a diagnostic platform also. I can use these techniques on the diagnostic platforms also. It means that to identify a disease also I can use it. A diagnostic platform, what I call it as. So I'm pinpointingly telling that the person is suffering with this type of a disease. Now, which type of it? I'm giving an examples of the SARS. This means that the COVID-19. We can find out the genetic disorders here. So before the birth of the child, when little bit amount of the fluid which can be extracted under the ultrasound guidance, I can take it from the what you call it as an amniotic sac and puncture the fetus and remove that amniotic fluids. And that fluid analysis can be done to find out the genetic disorders by using this technique called as an polymerase chain reactions. Very simple terminology. PCR we call it as. Let's forget about RT-PCR, qPCR, QRT-PCR. That we are going to have a discussion later. So that this polymerase chain reaction, which tells about the molecular part. Now, for example, I have a DNA, the DNA strand which has been present into my body, every DNA strand which has been present into my body like this. This is the DNA strand, what you can see here. But I've got it, the two chains here, the first chain and the second chain. And then these two chains are adopted. And then in between these two chains, I have certain nucleotides which is arranged in a sequence. You're going to learn in biochemistry when the molecular biology is being taught to you. Now, the presence of these nucleotides and arrangement of the nucleotides in between two strands. Now, this will tell about the gene capability, whether the gene 1 or a gene 2, which type of a gene. So the arrangement of these particular nucleotides between these two strands will tell about the type of the genes. You're clear with the concept what I'm telling to you? Okay. Am I fast? I am. It's okay. The pace is okay. The pace is okay. Perfect. Oh, that's great. That's great. Now, how do I use this? As I am discussing with you, that the diagnostic platform I can use this technique, which we call it as a molecular technique, call it as in polymerase chain reactions. And then after that, I can use as a prognostic test also. Now, I have a regime of the treatment. The regime of the treatment which I am giving you for the COVID now, it's a blind treatment what I am giving you. I am giving an antibiotic, I am giving an steroid, I am giving an antihistamine, I am giving one calcium dose, I am giving a vitamin dose. I don't know which is working onto that. Now, the prognostic value. Now, I wanted to know the line of the treatment which has been done by me vaguely. Is that is decreasing the load of the virus in the body or not? So when I wanted to find out that, I need to depend upon certain molecular techniques, what I call it as a polymerase chain reaction. So now these molecular techniques will play a very important role in the medicine, either in the diagnostic medicine or in the prognostic medicine. So the diagnosis and prognosis I'm purely depend upon. Now, as these techniques have evolved in the world, the mortality we are able to decrease it. Because I never had an idea that what is the virus and which type of the sequence it have, which type of the nucleic acid it presents here, whether it is the RNA dependent, or which RNA, what is the sequence of that particular. So any antigen, I can have the final molecular analysis. When I have that final molecular analysis, for me very easy to develop a drug, develop a vaccine, develop an antidote for that. So it gives an importance for us. So that when I'm interpreting this particular type of a results, whether I say positive or a negative, positive or the negative, it's very important for me. It is very important for me to look at and do the analysis of that particular report, what it is presented to me. Got the point here? Now, when I look at this report, which is there in front of your screens, now there are one value which I call it as a prognosis chart. Did you see here what we call it as a prognosis chart here? Now, then it has given this CT value. We call it as a cycle threshold. We call it as in CT value. Cycle threshold. What do you mean by a cycle? It means that an event which starts, events, for example, the event which is starting with the decoding of a DNA, the DNA is being decoded here, and then I have this cycle continues for the formation of the new DNA. I call it as a cycle. It means that the one cycle. What is that? We are going to learn it. So this we call as a cycle threshold. Now, the cycle threshold which is shown here, it is completely depend upon the load of that particular antigen. Load of that particular antigen. Keep in mind, it's a very important point we are going to discuss here. The load of that particular antigen. Now, the load of the antigen and cycles are inversely proportional. That one point we need to keep in mind. What does it mean by the inversely proportional? Whenever I have a load more, the virus which invaded or the antigen which invaded my body has been more, the cycles what I detect, I do the cycles for the detection and diagnosis of that particular antigen. CT, it means that the cycle threshold will be lesser. Whenever cycle threshold is been lesser, I have the more load. I have got the more load of that particular antigen. In this case, we call it this one as an antigen. We termed it as a COVID-19. Now, once again, I have the mid CT value, which gives a very importance for me. Say that a drug regime is been followed for a person for a week. Then I have made in them test that. So I can find out the load of the virus, whether it has been decreased or not. So the cycle threshold, which we call it as a CT value, will give a very important clue to a physician. A treating physician gives an important clue that should I decrease the standard of the dose? Should I have a change of the regime of the drug which I am using it here? Then high value of the CT and the very high CT values, and if we call it as a negative, we call it as a negative. Now we'll have a discussion once again when we understand all this process. I have this slide presented to you once again once you understand all these things here. Now, when I see at this bottom, when I see the left bottom of my cycle where I'm putting my stylus here, that this is what you can see here. These are the graphical representation of quantitative measurement. I'm doing a quantitative measurement here. So what do you mean by a quantitative measurement here? That we are going to learn in little more time. So the quantitative measurement will tell me about that this is the fluorescent technique or the fluorescent probes or the fluorescent material which will tell me, because when I see the virus, it don't have any color. So when it don't have any color, I need to add it to give it some color. So I use some dyes, the fluorescent dyes. I call it as a quencher. I call it as a reporter. I'll tell you that within a span of 1 or 2 minutes. So this particular dice will give me the quantity so that I can plot a graph and use this x-axis and y-axis and the y-axis here. So the concentration of this particular dyes, which have taken this particular antigen in a repeated cycles of, we call it as a cycle threshold, will allow us to find out the load of the virus. So I can have a quantitative measurement. I can have a quantitative measurement. This is a very important point here. Now, here you can see here the coronavirus is being placed on the right side here when I'm putting my stylus. The red color which has been shown here. Once again, the dice here, where it is been normal, there will be one baseline. We call it as the movement above the baseline, about decrease the baseline. Once again, a big story that is not required for you, but the basic points I am going to teach you here. So keep in mind that we are not going to do any mistake when are identifying or reading this PCR values, which we call it as in CT values for one particular patient who's suffering with the COVID-19. Now, on the right side and upper column here, where I am putting my stylus and rounding up here at this particular point, it tells me about some values. Once again, this is very important for me that when I was going through with that, the different types of these molecular techniques here, some people they come that they have come to a conclusion that the CT value of the 24, decrease of the CT value, the person is more affected, increase of the CT value, the person is moderately affected, or we can say some negative. It means that depend upon the reagents I am using it, depend upon that particular reagent, the value is going to be changed. Especially when I see this report here, they have given one, it is that the greater than 35. They have a value depend upon their reagent kit with what they have used is in 35. So that for me it's very important always to read this point at this level where I am showing to you this point here. So when I don't know about a normal value, what is the cutoff percentage when I don't know? It's very difficult for me to tell that the person is negative and the positive. So keep in mind, it's a fundamental point I need to understand when I learn about the laboratory medicine or a diagnostic medicine, which is play a very important role in identification of any diseases. I should know the cutoff value is what for that particular reagent. Same in the presence of a normal glucose levels here. We have the different method we are going to use it. You could have learned in biochemistry. So every kit have a different value. It is not universal that is 90 to 110 milligram. It is the glucose levels which is fasting. No, it depends. Depend upon the reagents what I am using, method what I am using. So keep in mind glucose oxidase method, hexokinase method what we have, it gives a different values. The same way that this laboratory report which is being generated here, you can see here the positive, the CT value is been 90. So this I call it as an analysis of this particular report. So I need not stick on to a one English word, call it as a positive or negative. You got it here. So this is how I have to do an analysis to have an approach for the treatment, whether it is for diagnostic or I'm using it for the prognostic purpose. You are clear here. Do you have any doubts that I am
Sir?
I have presented very clearly this particular points where you have to look to read a report? Then we'll go into the technique.
Sir?
Yes, tell me.
Sir, what if the value is exactly 35, sir?
So now see here, what happens? It is less than 35. Did you see the point here? You got the point
Yes,
here. They
sir.
have not given 35 equal or less than.
Yes, sir, that's what. If it is
Okay.
exactly
If it
35,
is, yeah, if it is
will it
more
be
than,
positive or negative?
see here, the cycles which is purely depend upon that when we say 35, it is, no, this is a greater than 35. What does it mean by a greater than 35? It means that the 36. You got it here.
Yes, sir.
So we say, see here, when I see here 1, 2, 3, and 4 points, you can see at this particular prognostic chart. Did you see the prognostic chart here? The person which is
Yes.
affected has given a drug regime for the treatment. Then when they have repeated the PCR here, he have shown the value of 24. Don't you think that the prognosis has been done by that particular patient? The load of the virus has come down.
Yes, sir.
You got the
It's
point here.
decreased.
Okay, so the 35, it is greater, sir. The greater than 35 and less than 35. The point I call it as less than 35. So I can name them that whether they are severely affected, mildly affected, or high CT value means it is more than that, more than that. It means that which is near to 35, near to 35. So most of the time, I need to understand that diagnostic values when we use it, we use this terminology called it as whether it has been greater or lesser, any value greater or lesser. In this case, it is that the greater and lesser than 35 value, greater than 35 value. 35 keeping it as a constant, as a constant. Got it here? So you
Yes,
say that a
sir.
person said value is 35, 35 is his value. Another person is 18 value. Now your doubt is whether should I take this 35, consider that fellow is positive or negative.
Yes, sir. That's my
Got
doubt.
the point here? Yes or no?
Yes, sir.
Okay. When I look at this point, is they could have used one term called it as an equal to and less than and equal to 35. Got it?
Okay, sir.
Okay.
Sir, so 35 is considered as positive.
Yeah, it's considered as positive.
Thank you, sir.
Okay. So this is how. So we can use it in the prognostic part also when the drug regime is being started with them, a treatment is being given to them. Clear? So that's why when I see here, this report which is being generated here, it will tell me about visit 1, visit 2, visit 3, visit 4, visit 5, visit 6. So all these visits, when the person makes it visits and the sample is being collected, I will plot it stating that I can use it for the research purpose, stating that this drug regime has been worked, the load of the virus has been decreased starting from the first visit to the sixth visit. I can find it out. And after the vaccination, the dose of the vaccine, it also helps me about the dose of the vaccination. Say that the person have taken the first dose, what is the load? I can find it out. The second dose, what is the load? Then the person when he take the booster load, what is the load? Then originally when he was infected directly, that time what is the load? It means that I can have an analysis and my body immune response also I can find out by using this visit chart, what I call it as visit chart, what we are going to call it as. So hope you have understand at least the basic points of that what I have presented to you here. Can I proceed on to the PCR? Okay, fine. Let's go to the main topic, what we call it. This, till now we have read about. What is this for? PCR, what do you stand? What is used for? Where I am going to use it here? Now, I have to study about environmental microbiology. Now, I wanted to find out that there are certain bacteria which are good for me. I call it as a probiotic. So I am, Yakult drinks we are making it here. So we are drinking here because stating that no, the Yakult is very good for me. Advertisement in the TV. and all, you are going to see that. So environmental microbiology, which are the good sources of the good bacteria for me? Now, till now, I used to think that the curd will have a very good bacteria. Yes or no? Now, I get the sachet, the persons who are suffering with the diarrhea, the loose motion for the longer duration, they give the sachets of lactobacillus, which is a recombinant technology. They have used it, made this. So that I need this polymerase chain reaction to be developed because I should know the genetic sequence of that particular antigen, that particular substance or bacteria or the virus, which I am making it artificially, artificial intelligence I am using it. So I can say that the PCR technique, which we call it as polymerase chain reaction, is a technique of an artificial intelligence. When I made this statement, I support my statement with the scientific evidence later. Now, what we are going to do in the genetic research here. Now, when I see here the genetic research, which plays a very important role, that now we have a strain that now when we read about the religious scriptures, when we read about any scientific picture, they put was tree, we call it as a family tree. So when I want to trace my family tree, so my genetic research from me. Now, if you could have watched one movie called as The Sixth Sense by that hero, I forgot his name of the hero. What is that hero? Surya. You remember that he some Bodhidharma, one person will go to China and that beautiful Shruti Hassan, no? That lady will go and collect his sample and then she will trace his ancestors which are from India, but he had that gene, that's why he has developed so many things. So when I wanted to do a genetic research, I need to understand about polymerase chain reaction. Then what is the another point we need to understand? That we need to understand that I need to find out and forensic evidence. How do I support? Now, recently I was watching some files in the channels of Discovery, so that there was one murder which was done. They found one body at one plant, one creeper. The creeper is cotyledon. So the police people, the forensic people have got evidence that the cotyledon is the one which will give the clue that who is the suspect. So the car which is parked in the bay, house bay, her own house, her own husband. So they got that cotyledon bean the back of that carry truck. And then that bean was matched with the bean where the body was found. And they found out who's the culprit. The husband was the culprit. And then they sentenced him to death. So that the forensic evidences I can collect it. The forensic evidences I can collect it. So it's a scientific based medicine or the scientific based, what I call it as a procedure which tells about the forensic evidences so that the persons are culprits. Forensic part also I am going to use it. Or a drop of the blood which I found it here. I wanted to find out that this drop of the blood is belonged to a who, or a culprit, or the person who has been murdered, or a murderer. So I can have a forensic evidence onto that. Then another which is very important point for me need to understand about food and agriculture. Now, as the population is growing here, I cannot wait for the crops to grow for a year. I need the crops to come down to 6 months and give me a big productions. So when I give some big production, I can supply to all the people. So I use this recombinant DNA technology. So when I want to use this recombinant DNA technology, I need the polymerase chain reaction to identify that particular the sequence of the nucleotides where I can have a synthesis of new DNA. New DNA. So that's what it is very important for me to understand about. So the food and agriculture is also. Then phylogenetics also know from where the human have come from this vertebrates, where it is the whole the phylogenetic structure I can find it out. And then for us, which is very important, is that particular point what we call it as in medicine. We had a discussion onto that. So that the medicine, any medicine I am preparing it here, So that vaccines I am going to prepare or any medicine which I am going to prepare here and antidotes which is being prepared by me have a very important role. We call it as a polymerase chain reaction. Polymerase chain reaction. Now see here that the consumer genomics. Now I need genomics and the consumer genomics I'm going to use it. So it means that this is the whole function of where I can use my PCR. It means that polymerase chain reaction. Now, I can use in the environmental microbiology to isolate some good bacteria. I can use it in the medicine. And phylogenetic, I need to find genetic research. I wanted to find out who are my ancestors. Forensic science, I am going to use it here. Food and agriculture, this is a very important point so that the growing population needs a more amount of food to be served. How do I get it? I don't want 1 year crop to go, 3 year crop to come here. So I give genetic modifications of the disease. We have a good lab which is opened by, I think, George W. Bush, a small one fellow, who has come, not Bill Clinton, which has come to Acharya N.G. Ranga University. In olden days, it has been called. We have a good DNA center there where the polymerase chain reactions will go very nicely. Anyhow, so the consumer genomics is there, and cosmetics, what I am going to prepare it here. So each and everything here is a big role which is played by polymerase chain reaction. So it is very important for me to learn the technique and how it is going to be used. What are the fundamental steps in polymerase chain reaction? Now, see here. What we are going to learn in this, we are going to learn about the basic concepts of the PCR, then how we are going to find out the steps in the PCR. Then it's very important, we call it as a QRT polymerase chain reaction. Quantitative measurement also we can take it to find out the load of that particular virus, especially hepatitis B, COVID, where the quantitative measurement will tell about the load of the virus and how much it can damage our body, and also helps in the prognostic description. Then I have the fluorescent dyes. Which type of the fluorescent dyes? Because I am doing a quantitative method. So when I'm using a quantitative method, I need some dyes to be used, and the probes also should be there, so that when I have these two strands here, so that I will take one probe which is been like this, the reporter and the quencher, what we call it as. This is an artificially made probes. The probes will come here, the primer will be given, and the polymerase enzyme will be used for the synthesis of these particular nucleotides. So the dyes and the probes, polymerase probes, which are very important for us to have a sequence of that particular nucleotide to be placed in that particular part against that nucleotide. Like I have a guanine, which is cytosine. I have an A, a T, or a U, a A. So this sequence which is placed, you learn in molecular technology, could have some knowledge in CBSE and even the intermediate level also. Then we have some primers. So these are designed artificially, PCR primers, what we call it as. Now, when you want this primer to be designed, I need certain softwares. It's a very simple technique, we call it as a softwares. The software is going to design that primers. Now, how is going to design? It's a big story. Once again, that's not required for you at this point of time. Part by part, you can develop by reading the books, and self reading is very important for me to increase my knowledge. So these are the software. I give you some examples of the softwares which are used to design these primer softwares. Now, next point. What do you mean by let read about a little bit of an history that went from the started here. In 1984 itself, 1984, maybe I was approximately 10 years old. So American biochemist, this fellow in our American biochemist, we have used this, call it as a PCR, is now the Nobel Prize has been awarded for this, both the people, Japan Prize, Nobel Prize is awarded to them 1993. It means that way back in 1984, when I was 10 years old, the techniques has come into evolution. Now, part by part, they started adding to that, and then the technique has come in 21 in the form of a good package to us. So this is an history which has been for the polymerase chain reaction, which has been developed. Now, however, the principle of replicating the piece of an DNA using two primers had already been described by Our own scientists in 1971, before I was born, 1971, such a beautiful scientist, we call it as the father of the genetics, Gobind Khurana, he was our Indian, has done this job of replicating the DNA using two probes. The same technique we are going to learn it here today. Two probes we are going to use with the help of the polymerase enzyme, which helps in the arrangement of the nucleotides between two strands. So that with the two polymers, I have a DNA strand here. So one polymer is going to synthesize, another polymer is going to synthesize. So I get from one, I get the two DNA fragments. From two, I get the four DNA. From four, I get the eight DNA. From eight, I get the 16 DNA. From 16, I get the 13. So on, I'm going to get millions of that particular fragments of the DNAs. Got the point here? It's very important for us to understand this particular point here. So this is mainly by using two probes. Way back in 1971 only that the Hargobind Khurana have done this job. The using primers, synthesizing the new DNA, synthesizing the new DNA. Now, what does it mean by this polymerase chain reaction? Is a powerful technique that has rapidly become one of the most widely used technique in molecular biology because of its inexpensive and simple. The procedure is simple. Machinery, what we used to in olden days, I'll show you some machinery which are used to be in olden days. The new days, we have a movable machinery, pocket friendly machinery, what we call it as. So we bought that tests, genetic test, molecular basis test at the level of the lower level, so that the person without money also can afford to go for the diagnostics to identify the different type of diseases. This is why we are using this PCR. We call it as a polymerase chain reaction. As the name implies, it is a reaction. Polymerase chain reaction using a polymerase. It's a chain continuation of the cycle here. Cycle 1, the cycle 2, cycle 3, cycle 4, and it is a reaction. Now it's a cascade type of a reaction. So definitely I call it as the name itself will tell me about it's a chain reaction using a polymerase. So I call it as a polymerase chain reaction. So one DNA molecule, as we had a discussion in the last slide, that it can produce the two copies of that, and the eight then fold. It means that it multiplies to gives rise to a plenty of copies. You know, DNA, I got one DNA. From one DNA, I can generate the millions of the DNA by using this technique called as a polymerase chain reaction. Now, this continues of a doubling. This continues of a doubling. A specific protein known as a polymerase, an enzyme. This particular enzyme, when you use this polymerase enzyme, polymerases, by using this polymerase enzyme, what it does that it will allow the sequence of, as I told you that this is the two strands here. The polymerase here, the probe is going to be synthesized. Another probe I have got it. The probe comes and attached with the repeater secture and the quencher secture here. The polymerase which is been present here is going to replace all the nucleotides in the sequential manner to give it to me a one DNA molecule is replicated or amplified into two molecules. Then once again I have got the two DNA molecules here. Then I use these two probes and polymerase enzymes so that it gives me a four molecules. It gives me a four molecules. So I keep on synthesizing it. Now, together, so what it is, an enzymes that are able to string together the individual DNA building block to form a long molecular strands. So I can find out the sequence of that particular nucleotides. To do the job, polymerase requires to do this job for an amplification job. What I speak, I use a term called as an amplification. To do this job, what I need, polymerase requires the supply of the DNA building blocks. What are the DNA building blocks here? The nucleotides consisting of four bases. I have a basis, what I call it as thymine, cytosine, guanosine, You got the idea? The thymine, the cytosine, adenine. So all these nucleotides is been required and it should be supplemented when I do an experiment. There's a theory part which is attached behind. We had a discussion about in the previous slide we have discussed. Now, what I need when I want to initiate the PCR, what I need. So we call it as a components of the PCR. So when I think of a component, when I want to start my PCR, definitely I need to get a DNA sample. How do I get a DNA sample? I need to get a DNA sample. I need to separate the DNA with the molecular techniques. What are the molecular techniques? Say that a tissue is being given to me. A tissue of the liver will be given to me. With the biopsy, the people will take some tissue and given to me, or a sample of the blood will be given to me. I need that particular DNA. I wanted to find out like PSAG. I wanted to find out from the nasal swab. I want to find out COVID-19. When I take the nasal swab, plenty of the tissue of my nose from the nasal concha also will come. Plenty of epithelial tissue also will come. How do I know that the tissue is belong or DNA belong to the COVID-19 only? How do I belong that the tissue which is given in the form of the liver, the biopsy tissue, or in the form of the ovary, it have a special other than the other than the body DNA, it have got some other infected DNAs. It means that the viruses or any antigen which is invading my body. So we use a mortar. We put a little bit of buffers into that, and we do grind it. Generally, how we grind our masala, no? The same way we are going to grind it here. We extract the tissue fluid from there. Once we extract the tissue fluid here, we keep it in the centrifugation technique methods, and depend upon the centrifugation by using an auto pipette, we are going to filter and remove the DNA. It's a big procedure once again. Keep in mind what I need it. First, I need the most important point that I call it as a DNA sample. I need a DNA sample because it is polymerase chain reaction. Got the point here. Next, I need certain primers. What I am looking for? No, what is this particular primers? Primers are the one which will allow the synthesis of that sequence. The sequence. Now, A should be placed. Now, here I need a T to be placed. For example, G is been there. Now, I need the C to place. When this is a two strands, this strand is completely different. I want the nucleotide to be placed here. I want an opposite, this nucleotide to be placed here. This is what I call it as an probes. What I call it as an probes. Then I need certain nucleotides because it is a machine. Now the machine needs certain things so that if I give only, then it will help to synthesis of the new DNA molecules. It helps in the amplification of that particular molecules. I have to give these nucleotides. And these nucleotides is being given with the help of a tube called it as a PCR tube. That what we call it as an aliquates. We have a test tubes. A smaller one we call it as cuvettes. And it's a smaller one we call it as an aliquates. These are the silicon one which does not interfere with any reaction. Like how we have the syringes nowadays, this one. It is made up of the same material. It does not alter any property of the fluid which is being present. It's a very important point. Now, see that the container which I am using it here, if it is affects the property of that particular solution or that particular fluid, it leads me a bias. So I should use it so that the PCR tubes are called as an aliquates are made for only for PCR, a standard tubes which will be supplied by the company. So that I should not have any biotic studies. Then I need a buffer, the buffer which has been very important. So that the nucleotides I am supplying, the primer what I am supplying it here. So those primers and nucleotides, everything, these primers and nucleotides has to be mixed into the buffer, such as a phosphate buffer. You'll come to know when you go to the molecular pathology when you study in the second year. So even in the tissue culture, we are going to use this phosphate buffer, very important role here. So we have some specialized buffers. Then very important one is TAQ polymerase. Now, this TAQ polymerase is the one which is going to initiate this particular formation of these nucleotides, the new chain formation from the DNA. Clear? So it means that the fragments of the DNA, what I may have, from that I am going to new DNA. But one more important point I would like to tell you. Now, our viruses are plenty of them are in RNAs. So even when I have this RNA, I can decode the DNA. That I call it as an reverse transcription PCR. Got the point here? Real-time PCR is a machine. The process I am using it to identify, call it as a reverse transcriptase. Why? I have only RNA molecules, it's single strand. It's a single strand. The virus is being a single strand. I need to have a DNA of that particular virus. I can synthesize that using a reverse transcriptase. Here I am not going to use the polymerase when I run about an RT-PCR, reverse transcriptase PCR. I use a reverse transcript polymerase enzyme. I am going to got the point. Reverse transcriptase enzyme. I am going to use it when I run RT-PCR, not real-time PCR. The name is real-time PCR. Don't get confused. Okay. When I'm extracting DNA from DNA, it's okay. From RNA from DNA, I have to use this reverse transcriptase, which in the case of COVID-19 or any viral particle, when I wanted to find it out, when I prepare a vaccine for the viral, I have to decode this mRNA, messenger RNA. That's very important for me. You need to keep in mind. Now, what it is. It have got the different cycles. You see the different cycle. Now, this is the DNA strand which is being given to me, the DNA strand which is being given to me. I want this DNA strand to get fragmented, broken down. I want this DNA fragment to be denatured. So, there's a first step what I'm going to use called as denaturation. Now, this particular denaturation, for me, it is very important. One minute, let me take out these notions. Gosh. Okay. Once it is been denatured, where I have to definitely temperature. Now, when I'm eating the protein, generally what happens that nowadays scenario is very bad. I go to the market, buy little amount of the chicken, throw it into my freezer. Then I remove that when I want to cook it in the small pockets, I keep it. So that the small pockets I throw into my microwave. Then I use the defrost technique. So freezing and thawing also will do the denaturation of the proteins and the increase of the temperature. So that this what I call it as a thermal cycler. Though, so whatever the samples I am going to put it here with the help of and using of an liquids here, that the thermal cycler, it means that this liquid, the PCR tube, it is placed in this thermal cycler, will increase the temperature to 95 degrees. So at 95 degrees, it means that a specific protein or specific DNA molecule will have one set temperature of denaturation. So when we have the set temperature of denaturation, in this case, we are use the DNA here. I can do the denaturation of the DNA at the level of 95 degrees. At the level of 95 degrees. Got the point here? So this is very important for us to understand. Next, what happens here? No, I need the primers to bind the templates. Now, once the denaturation is been over, I have the formation of the one template and the second template is been formed. So I have a process called as an annealing. So because once I have these fragments, which is been, this is a DNA molecule, it's been separated from here. So like this, there's a DNA molecule which is attached with the hydrophobic bands bonds here. So now this has been detached as a single fragment here, a single fragment and the single fragment. I want the polymerase enzyme, this enzyme, one polymerase to come and attach another polymerase. So these polymers, we call it as a templates to come and attach to these basic template molecules and keep on synthesizing and keeping this, what we call it as a nucleotide, to give rise to me a new strand of a DNA, new strand of a new strand of a DNA, new. So I had one, I had two in the second one. Then the 2 will become 4, the 4 will become 16, the 4 will become 32. So like that, I have an amplification of this particular strands of a DNA. So for that, temperature also is important. When I'm treating it here, it's very important for me that I need to maintain the temperature of 55 degrees. If it is more, I can't do. If it is lesser than this, I can't do, because annealing is nothing but the primer is going to attach onto the templates of the DNA, templates of the DNA. So this is very important point we are going to learn. So what is the basic point is nothing but the variations of the temperature. Who will take care of the variations of the temperature? It is the thermal cycler, what which is being used in the technique of an PCR, the whole small type of a machine, where I keep my liquids in the wells we call the wells. Now, after the annealing, the third which is nothing but an extension. You can give here that the primers is going to get attached, the primer is going to get attached. Say that example I have got A here, the primer will give T. Here if it is G, it gives C. Keep on the nucleotide in an opposite direction as it has to be arranged. So two primers, one primer for this, the second primer for this, it gives us a template. One template will be turned into a one DNA, and the second template will be turned into a second DNA. Then the same, I have got a two. First I have two. One DNA is turned into two DNA. The two will become four DNA. The four will become 16 DNA. So the fluid is going to get millions and millions of cycles. You get the point here. So, what do you mean by in one cycle? Got it? We have heard cycle. So, this cycle is nothing but for one formation. From one DNA, I've got the two. This is called as in one cycle. One cycle will have denaturation. One cycle will have annealing. One cycle will have an extension followed with the formation of this particular DNA. So now from one single, so I have got the one DNA and one it is been two. So formation of these two DNA molecules, I call it as a completion of one cycle. Got the point here? So like that cycles, it will have the formation. That's why the number is 1, 2, 3, 4, 5 cycle threshold is being given. So this is very important for us to understand. So one cycle, how many parts it have? It have a denaturation of that particular protein or a DNA molecule. In the case here, it's a DNA molecule. Then annealing, so that I have given the nucleotides which is been supplemented by me. The primers also is supplemented by me, and polymerase also is supplemented by me. So this helps in the synthesis of this particular new DNA fragments. So extension. So these 3 points, I call it as in one cycle. Then the two molecules what I have got in my fluid undergo denaturation to gives rise to 4. Then the 4, what it gives, that 4 one cycles, keep on we are recycling it to get more amount. Then it will be compared with what we have it, that I'll tell you later. Got the point here? Did you get it? Do you have any doubts here, guys, about the components and PCR process? So at here, you can see this recorded as a one cycle. Do I? Okay, that's great. Okay. Now, we have got plenty. So there are different techniques we use it. We have one step. We have two-step PCRs. Got the point here? So now mRNA is the see here. mRNA. So when I want to synthesize the DNA, I was telling you that I can have a synthesis of DNA from an mRNA molecules here. So the messenger RNA which I am going to. So messenger RNA. So what it use? I use an enzyme called it as a reverse transcriptase. That we call it as a reverse transcription enzyme. So reverse transcription enzyme, what it use that it's going to synthesize the DNA fragments. Then in the RC-PCR cycle, it will have an amplification of that. I'm synthesizing a DNA. Once I have a formation of this DNA, Then I have the same PCR cycle number 2. You're getting the point here? That I don't have a DNA molecule. I have a single fragment of an mRNA, messenger RNA. You know the messenger RNA, we have a transfer RNA, the ribosomal antibody I need in molecular biology. So this particular one molecule of an mRNA which I got it. So this mRNA molecule will allow me to synthesize the DNA of that particular antigen or an organism How do I? I use an enzyme called as a reverse transcription method. I use an enzyme called reverse transcriptase. Then I will synthesize the second strand. Now a DNA, when I have a DNA one strand, the second strand, I have all the nucleotides attached here. So reverse transcriptase I am going to use from this direction, and then this reverse transcription is going to put its base pairs. Well, this is a completely new one which I am synthesizing, which I call base pair. And then the new DNA molecule, for me it is very easy. DNA of that particular organism is this. For me it is very easy to prepare a vaccine. Very easy for prepare any diagnostic test. Very easy to prepare the load of that particular. For example, a virus which invaded my body called as an hepatitis virus, it have a messenger RNA. So when I do this PCR, polymerase chain reaction, which I am using it here, it is a DNA basis. I want the DNA to have the repeated cycles to identify it. But the virus does not have a DNA, it's an RNA molecule. How do I prepare it? I prepare by a technique called as a reverse transcription. A very important point we had learned today. So I use one step also, I use two steps also. You got the point here. Now here it's very important for me to understand two steps also I am going to and one step also. So what I am going to here, I am using the specific primers here. Did you see here an addition here? Two steps. Got it here. So I vaguely think might be the tissue which is given to me is a liver. I am looking for hepatitis virus. I have some primers according to the virus. I throw that into the fluid of an liquid. Got the point here? Because I am looking. How do I look? Because the physician, depend upon his signs and symptoms, he will think that the person is suffering with the viral hepatitis. So which type of a virus? It might be an HBsAg. So that this hepatitis virus, born A, B, or C is a different issue. But I know with the clinical symptoms. So when they write that, expected this, so we use this primer in the diagnostic part. So that for me it is very easy. Because the result which can be generated in us, this result by using the two-step RT-PCR, real-time PCR, or reverse transcripted PCR, which I am using it here, this can be done in minutes. So that diagnosis I will reach faster. That's the importance of this one-step and two-step PCR techniques. Got the point here? Hope you guys are fine and you have understood here. Since the time is not permitting to continue here, so I stop here. Then I'll be taking the next class maybe tomorrow or day after tomorrow. So keep in mind that I have taught you till one step and two step. It's very important things we are going to learn from now onwards. So try to attend and try to be present here in my class. Did you understand? Did you understand here? Anyone else with the microphone on? Can you speak to me?
Yes, sir.
Okay.
Yes, sir.
What did we learn? Very good that being a medico, I wear a white apron. Any one of my relatives or anyone comes to me, when they give me a PCR report in my hand, I don't stick on to those English words, nonsense English word called positive and negative. You got it? So I read the report. So I told you what you have to look to read the report. Then we had come to the PCR technique like that. So what we have read up very beautifully about the components of the PCR, what I am going to use. And then what do you mean by primer? What do you mean by a probe? How it is help in the synthesis of the nucleotide base phase? What do you mean by amplification? Also, we have done it in a beautiful way. From past 1 hour, we were able to explore the different types of the PCR techniques, which we call it as a two-step PCR and one-step PCR. Polymerase chain reaction, with the DNA, how do I synthesize? Also, I have learned with the single mRNA strand how I am going to synthesize. Also, I have learned it. And then having a DNA, this is how I am utilizing into the treatment aspects. So it helps in the diagnosis, it helps in the prognosis. What I call as the importance of the polymerase chain reaction in the medicine in other ways, such as genomics, some genetic disorders to identify all those things we have learned. Great, great guys. Thank you so much. Because I think Saileja madam is waiting for the to join. Dr. Saileja, you can join, please. Thank you so much for your patience for 5 minutes. I'll
Thank
catch
you,
you
sir.
tomorrow, guys. Have a good time and enjoy the days and read well.